I'd like to introduce our next presentation. We have a double team in Kate Comstock, who is the Life Science Mass Spec Senior Marketing Specialist, and Kyle De Silva, who is the Pharma and Biopharma Marketing Manager.
A slightly provocative title, No More Unknowns. What we are going to be presenting to you today is some new technologies and workflows that have never before been used for extractable and leachable analysis that we believe can give greater insight into the identification of unknowns in very simple workflows. So, three parts really to the presentation today. I'm going to show you a case study where we looked at some pharmaceutical grade O-rings with a whole suite of different technologies and capabilities. And I'm going to introduce some brand new technology that's never been used, as I mentioned. And my colleague Kate is going to demonstrate the capabilities and advantages of high-resolution, accurate mass spectrometry for the analysis and identification of unknown extractable impurities. So I won't go into any background on extractables and leachables. I'm sure there are many people in the audience who have forgotten far more about extractables than I will ever know. But I think the one thing is key from all of the industry body guidance and regulatory guidance is that you really do require a suite of analytical technologies to complement and identify extractables, which can take various different chemical forms. So in general, there is a requirement for volatile analysis, semi-volatile analysis, non-volatiles and elemental impurity analysis. And I think it's fairly safe to say that conventionally, mass spectrometry has really led the way in identifying unknowns. And as instrument vendors, we recognize that many people within the industry have reached out to us in the past requiring additional capabilities. So there has been a general movement from mass spectrometry to high-resolution mass spectrometry, both for LC and GC amenable compounds, often complemented with orthogonal detection such as UV, or more frequently today we see charged aerosol detection as a universal detector for unknowns. So we're going to present two or three of these analytical technologies today, but it's fair to say that beyond the analytical technologies, there are requirements for additional peripheral technology to support extractables and leachables workflows, and we can support that right the way through from the extraction step all the way through to the very ultra-pure reagents and vials that really give you a zero-level background when performing your analytical test. Okay, so to take a look at the case study, we worked in collaboration with Smithers, who were able to provide us with a series of pharmaceutical-grade O-ring extracts that we then analysed with three different technologies. So for our non-volatiles analysis, I know many of the laboratories in the room are very familiar with our Orbitrap-based mass spectrometry technology, in particular the Exactive series instruments. These have been around for a number of years and have been used to great success for identifying unknowns. For the first time this year, we launched an instrument in June at ASMS. It's the brother, really, of the LC variant Exactive. It's a GC MS system with very unique capabilities. So we analysed all of the extracts using these three technologies, including quadrupole-based ICP-MS. So just a brief overview of what we did. We loosely followed the BPOG guidelines from last year. We took the O-rings, weighed them out into vials. We chose a subset, really just for brevity of the analytical process of solvents to use. It really is just a case study, not an exhaustive study. And we held them at elevated temperatures for 30 days and then analysed them. Fairly standard analytical process. Those familiar with GCMS will see that we used fairly typical testing conditions. I would say we operated the system up at 60,000 resolution, so this is a resolution that really can't be matched from any other technology on the market. But the system itself can go faster than that and at higher resolution than that. And then we process the data in a new automated data processing software. So what is the system? Well, it comprises three key established robust technologies that we brought together almost like Frankenstein into one nice package. And we have an established GC system, a truly modular system that allows multiple different injector modules to be dropped into the top, headspace, program temperature, vaporization, so on, all that great tech that people know and love. And then behind that, we have an ionization source that we developed for environmental analysis, actually. People who wanted to run robust sediments for weeks on end without ever having to clean their instrument. So this was the most robust source we've ever made. We're very proud of it, and we were sure to include it within this system. And then behind that, the Q Exactive high-resolution GC Orbitrap-based mass spectrometer. So inside the mass spec itself, we have a quadrupole that kicks ions into what we call a C-trap, and we can do a very elegant ion dance, and we can move them, select a mass if we want, or we let all the masses through. We store them up, and then we spit them into our Orbitrap to do the very high-resolution analysis. But we can dance them into what we call a HCD cell to do very complex and fragmentation experiments, and then kick them back into the Orbitrap. And we can move these through in an automated way, data directed and automated as the analysis proceeds. So what does the system give you? As I mentioned, it's 120,000 resolution, which seems an obscene number. But that kind of resolution allows you to really clean up spectra. You can deconvolve spectra and have a very clean spectra without any background ions. That means your library search is more effective and you get higher quality identifications. We also have an exceptional level of mass accuracy. I've got a few slides where I deep delve into this, but we in routine run at 1 ppm mass accuracy, and the value of that is explained in the next few slides. But in reality, what this means is you have less identification options and higher degree of confidence in your identification of your unknown. And we're able to do very, very low sensitivity analyses, so down from the very low femtogram up to the high nanogram in a single run. What does this mean? In reality, you don't need to dilute extracts to do a quantification experience of a high peak, but you can still identify a tiny little trace extractable in one run, unlike on other technologies where you have to set up a more elegant and complex analytical sequence. So this is a GC peak of an extractable from one of our O-rings, and what we've done is we've shown every single spectrum across that GC peak. So this is acquired at 60,000 resolution, and we can see we have plenty of scans across the peak, and every single scan is less than 1 ppm mass accuracy. So other high-resolution technologies, they would need to take an average across the peak to come up with a mass accuracy result. And that in itself introduces errors, whereas we can take any peak and have confidence in that mass accuracy result. So as I mentioned, it's exceptionally sensitive. So this is a test that it's a system suitability test. We can run it every day on every system and guarantee. So this is 10 femtograms on column. And we run that. You can see we never see any noise, so signal-to-noise experiment really doesn't make any sense. So what we just ask for is replicate analysis of this, and then you take a confidence limit. So we have a limit of detection on this system down in a very low single femtograms. And this is in full scan all the time, all the data all the time. So no requirement to do MRM-type experiments, even in high matrix. And again, as I mentioned, it's linear over a very wide range. So unlike TOF technology with ADC or TDC detectors, you'd normally see some form of saturation at the high end. We don't see that on this system at all. This is 10 femtograms, right the way up to 10 nanograms, perfectly linear. So effectively, that just means less analyses, more time in the lab. And the beauty of this is a mass accuracy plot over the same dynamic range. And we never leave that 1 ppm. And this is absolutely unheard of. Very, very high concentrated peaks, 1 ppm right across the range. So all of that technology is normally fairly complex, and we work very, very hard to ensure that it isn't. So from start up to analysis, it's 1 minute, and you can really set your clock by it. It performs all of the instrument setup, quality checks, leak checking, all of this kind of stuff with a single button on the system, so that you're off and running with very little user interference. Okay, so that's the system. Let's take a look at some of the data. So to analyze the data at Thermo Fisher Scientific, we've got a range of software suites dependent on customer requirements and needs. I'm going to be talking about Tracefinder, which comes as standard with our Exactive GC instrument. So this is both for quantitation and unknown ID, or Quanfirmation as we like to call it. My colleague Kate will talk a little bit about Compound Discoverer, and we have a very nice presentation scheduled this afternoon on a new cloud-based library that I won't go into any detail here. But all of these technologies can hook in together, and it depends on your analytical workflow for exactly what you need. Tracefinder is very simple to use. It's designed for the environmental market, for operators without a high degree of experience in mass spectrometry. So it performs an automated deconvolution of all the complex high-resolution mass spec data, and then gets an ultra-clean spectrum because of the high resolution, and does a library search, and then has some nice proprietary accurate mass filtering algorithms that give you more confidence in the result. So as I mentioned, deconvolve, clean the spectrum, nice clean spectrum, and identify the candidate from the library search. So one of the algorithms underneath the hood is what we call a HRF scoring algorithm. It's fairly simple, actually. It's a high-resolution filtering score. So when you identify the molecular ion, that gives you some information, but you've got all that nice fragment ion information that you don't want to lose, and that really gives you the confidence in the analysis. So all of these fragments also have exceptionally low mass error. So what we do is we take the proposed formulae from our library search, and we take a look at every peak that we find observed within the spectrum that can be explained by that proposed empirical formulae, and we divide it by all of the unexplained peaks. So it's quite simple, but the result of that means that we get a HRF score with a perfect score being 100, but combining that with the library fit, it gives an exceptional confidence in ID. So here's just an example of how the software works. Generally, you're running some kind of blank or standard or control with some internal standards. So here's just an example of our blank control from our O-rings and one of the results. I think it's fairly clear to see there were some extractables. So the software, as I mentioned, does an accurate mass deconvolution. So all of the ions are pulled out. They're found to, the retention times are all perfectly aligned, and those ions are built up into our spectrum, a nice clean spectrum. It then library searches that spectrum. But you can very easily compare series and batches of controls versus replicates of your sample. And it's very simple. We've not gone into complex, detailed principal component analysis. Customers just said, well, I have this bunch of samples. I want to compare it to this bunch of samples. Tell me the differences. So that's what we did. We heat mapped the differences out so that you can just go to the peaks that are maybe above your analytical evaluation threshold and identify just those peaks. So what you can do is you can rank the results by a fold difference to a blank or a control or a gold standard sample, and really just pull out the differences very quickly, numerically and visually from a heat map. So you end up with a result. You get a list of proposed matches and a high-resolution filtering score. So as I mentioned, what does that give you? Well, the library match here, the SI, if we take a look at this particular example, There were two fairly high library match scores, one on row 3, one on row 1. So on a nominal mass instrument, you'd have to do a bit more investigative work to work out which one was the true match. Maybe it involved some retention time data, maybe some pre-knowledge of the sample. But our HRF scores are very different because in this sample, in this analyte, sorry, there was spectral ions that weren't explainable by the proposed empirical formulae. Consequently, it has a low HRF score. The combination of these two gives a combined score that really would tell you that there's only one option for that analyte. And again, what we're able to do with MS that can be automated is you can take that molecular ion, you can fragment it up again if you want for absolute confidence in that. So here's a fragment ion spectra of that molecular ion with the results showing all of the mass errors significantly below 1 ppm. So what about when you don't have a library hit? So how do you identify an unknown without that additional backup resource of a library? So this happens quite frequently, but what you're able to do is use orthogonal ionization techniques very, very quickly. So we move from electron ionization to chemical ionization within a period of about 2 minutes, and this is real, you're often running. We run these samples together with Smithers, same day, same lab, within about half an hour of each other. So you're then able to take the CI data, and if you want, you can fragment that up again for additional information. So here's our EI spectrum of this unknown, molecular ion, and the molecular ion is proven with the formation of the adducts in CI. And then additionally, we can do fragment ion analysis of that CI molecular ion. So I've gone on about the mass accuracy for a long time and probably repeatedly, but this is the real true value because that mass accuracy on most instruments on the market would sit around 6 to maybe 10, some around 20 ppm on time of flight instrumentation down at these low molecular weights. In reality, at 1 ppm, the analysis of this unknown really would give you only one elemental composition. So within the period of a few seconds, you have a very high confidence elemental composition result to work with for an unknown that's not in your library. So I won't go into all of the peaks that we're able to identify, but there were numerous. We found a lot of stuff in these O-rings that are pharmaceutical grade, and we're going to put out a comprehensive series of reports on this. But fairly easily and with a high degree of confidence, we could rapidly identify a whole range. So then what we did is we took a look at the elemental analysis, so the metals within the same samples. So we took the samples and we analysed it by quadrupole-based ICP-MS. So this is our Thermo Scientific ICAP Q series ICP-MS instrument. So it comes together with a fully compliant software tool called Qtegra, and it also has inbuilt reporting and QA and QC checking capabilities that can be automated within the software for USP or the USP guidelines in draft for 232, 233 and the ICHQ Q3D guidelines. So we analysed these just by direct infusion. The 100% organic extracts we partially evaporated and made up with ultra-pure water. They were all acidified before analysis. The analysis itself, all of the QA/QC checks are automated within a sample list. So there's no user intervention to get this. It's a press a button and go. It tests all of your calibration range, the linear dynamic range, the sensitivity verification, the accuracy check, and it also monitors drift across the day to ensure that all of the QC criteria are reported for the data. It probably doesn't come as much as a surprise to many of you who are more familiar with this area than I. There wasn't a high degree of metals that were extracted from the O-rings, thankfully, but exceptionally low levels, we were, with a high degree of confidence, able to detect traces of antimony, of arsenic, of lead within them. They were all in the salt, high content salt extracts. There was very little in the organic extracts. And again, as I mentioned, we automated all of the drift checking and QC throughout the entire sequence of the day. The software kicks out an automated report. So with that, I'm going to pass to my colleague, Kate, who will go through some of our LC data.
I'm going to talk about the LC-MS analysis. Okay. I think what we are here, try to figure out that this, how do we do the ENL analysis better? I have to say, from my own experience, ENL analysis is become a mainstream and routine. We got a lot, requests from customer. I've been asked all the experts here because we are instrumental company, right, for all those tox study, all those, how to do the evaluation. It's out of, my knowledge, but, our customer keep asking us, so I can feel, right, that there are more and more ENL coming up. So I think we are all the, piece of this big puzzle. And for the instrument analysis, it's very important piece because based on that, you do your toxicology assessment and the eventual new drug application filing and put your product on the market. So I have to say, before I came here, we had a meeting with one of the arguably biggest, best pharmaceutical company. Just less than 2 years ago, they had the drug, very good cancer-fighting drug, but struck down by FDA. It's the stability issue. So at this point, they're not sure it's a formulation or it's leachable. So we're working with them. It's a shame, right? They spent so much time, effort, and now couldn't put this product on the market, So that tells us the high stakes for ENL analysis. I know a lot of you here are expert, but we just try to, review some of the basics. And as we all know, except the carbon-12, most elements, all the elements, their, atomic weights are now the integral, right? More than half elements on the periodic table have stable isotopes. Those stable isotopes have 3 unique properties. One, they have a unique atomic weight, right? How accurate depends on how many decimal points you want to use. And secondly, they have their unique relative abundance. Third, the mass difference to the most abundant is fixed. So these unique properties we call nature's fingerprint. Also, electron mass, right? Normally we don't think about it, but it's very important when you try to do your exact mass calculation. So for those hair splitting details, it's not our concern normally. But when you try to figure out the unknown structure of your extract leachable, the first thing you need to do is try to determine the right elemental composition. Those are the exact information you need, and those information can only be obtained by high-resolution mass spec. So this is actually, I borrowed this from our colleague, Alexander Makarov. So this is the analysis can be seen by different instruments, right? They are similar, but they are alike, but which one do you prefer? I think the answer is obvious. But most importantly, which one you have to have in order to get your job done right. So this is just a simple example for us a mixture of two small molecule compound at about the 1:3 ratio. At the 17.5 K, we can't separate, right? 35 K, we can partially separate. 70 K, baseline separated. At 140,000 resolution, they are complete baseline separated, also with sub-ppm mass accuracy. On top of that, you didn't sacrifice any intensity, which is very important when you do high-resolution quantitation, right? By the way, this is acquired by the Q-Exactive. We call Q-Exactive is a quant machine, right? You can do high-resolution quantitation very easily. Just by mass accuracy is not enough to arrive unique elemental composition, right? Because when you have a higher molecular weight than 400, 500, even though you have a 1 ppm mass error, the possible elemental composition can increase dramatically. Therefore, we need a second orthogonal filter to figure out the right, the unique elemental composition. So this is just an example. This compound pyrazine and has, we plot this the isotope distribution under different resolution, right? As we can see, only at the 240 K resolution you start to see all those fine fingerprint, right? This is very important. People always talk about the accurate mass. Sure, that's important. But when you deal with unknown, you have to have this. Remember isobaric, right? So, but this fingerprint is unique for one compound. So even though their molecular weight are very close, sometimes even almost exact the same, but those as fine isotope structures are different. So this is a real example we acquired. This is for impurity ID, right? I'm just trying to blow up this peak. And then if you just take a look at the A2 isotope peak, we can clearly see the split between sulfur 34 and carbon 13. But if you run this same example under 500,000 resolution, you can see more, right? You can see the nitrogen 15, oxygen 18, right? So that's why we say you have a high resolution mass spec, give you all those benefits, horizontally, what kind of elements present in your unknown in and out. And then vertically, by their relative abundance, you can calculate how many, right? So that's why we say you can do item counting, right? Okay, we are all too familiar with 391, but now all the 391 are the usual suspect, right? Only high resolution mass spec can tell. So this is actually the real problem I had when I worked at the Roche Palato. So we have this, right away I thought, oh yeah, this is the valley, right? The common LC background. Look at this dimer, look at this associated, right? But right away I said, oh, wait a minute, the mass doesn't match, right? So if we zoom in, take a look, right? This peak, actually, this pair, we have two pairs peaks. This pair are my impurity. And then the real valley is here. And also, I would like to draw your attention to this. This is the sulfur 34 peak. So, when we take a look at the isotope simulation, it's very clear. They are different, right? By combined with MS mass fragment, we quickly come up the structure. So I can't show you though. Okay, so this year is 10th anniversary of Orbitrap technology. Within these 10 years, this Orbitrap technology really, advanced leaps by leaps and bounces. So today's Orbitrap technology, the instrument we have a higher resolving power, is more sensitive, higher scan speed, mass algorithm, so on and so forth. On the other hand, the usability is up to, right? It's a small footprint benchtop. And we have a tone wizard, we have a method template. All you need to do is drag and drop, build up your method. And it's robust and basically plug and play, right? So this is Orbitrap mass spec we have, right? So fit all the budget and the applications. We just within the Q Exactive, like Kyle said, we have a series, right? So I will just quickly go through the analysis for the O-ring, right? I did the LCMS on the Q Exactive Plus, and it's very simple, right? We just shoot in and then got a whole bunch of data. The important thing I want to say here is the compound discovery 2.0. This is our own, okay, yeah, another thing I want to point out, sorry, is compound discover 2.0. We can do unknown impurity analysis very easy because we have the MD cloud and we have this homemade, we simply call it the mass list, which is common INL compounds database, and also you can do a ChemSpider search. So this is the mass spec method, very simple. This we call this a generic high resolution screening method. You just full scan top 3 MS mass, and then we can do polarity switching on the fly, right? And then you just drag drop all the icon here, and then specify the mass range and resolution you want. You're ready to go. Normally, data process is always a bottleneck, right? With the compound discoverer 2.0, bottleneck no more. This one is thermal made, yeah, with advanced, customizable, very flexible, workflow. And for the 1.0, we do no parent compound impurity ID. But for 2.0, we can do unknown, right? So which is great news for INL folks. And then in this one, we've incorporate the safe differential analysis algorithm. And also we have this wonderful Mass Frontier software which can do spectral interpretation through the fragmentation. That included in this software. We have MD Cloud, the only one high-resolution spectral database. So Robert, the CEO of ICAP, going to give us talk this afternoon. And also we have, like I said, we simply call it a mass list. This is some of the common INL compound we accumulated. Right now we have about 1,500. This is how the compound discovery look, right? On the left, you have all those nodes, right? You can just based on your need, drag and drop, put here, and then this is all your sample. So by the way, we did 4 different O-rings extracted used 4 different solvents, and then we did like a duplication. So everything is there, and then this is just show you the workflow. So this is a very powerful software. It's versatile, it's flexible, yet easy to use. Normally, when the software is powerful, it's hard to use. Not for compound discovery, right? And we have, if you don't want to spend the time at the beginning, you simply can use the build. We have a wizard there. All you need to do is follow the wizard, use the building template. Literally 15 minutes, you can start to process your data. Okay, sorry. Yeah, I want to say this. We have this ChemSpider and the mass list search and the MD Cloud search incorporate this. So this is excellent for the INL unknown impurity ID. Okay, this is how the result look like. We have different tables here, and we have the chromatogram and the mass spec, and look at the color. The color means whatever the identified compound, right, the software automatically calculate the isotope distribution. If within the 3 ppm window, if the mass is not right, we'll color code it in the red. In this case, I have green, so I'm okay, right? So this is, I was talking about, you have to have a spectral accuracy, right, or isotope fidelity. And for this particular experiment, you can view your result in whichever way you like in compound discover, right? This is the, orange A in 4 different solutions. I want to see how they behave in different solutions. See, right away I can tell, right? Or I want to see all the orange behave in one extraction solution, 50% ethanol in this case. Here you go, right? And also, of course, we have the PCA, whatever. So those are all on your finger clip. So this is the result for the MD cloud, right? Give you the list when you click the compound, and give you the mirror plot. On the top is the experimental result. This is the reference spectra in the database. So right away you can evaluate, right? And if you click on the MD Cloud ID number, and we'll bring the MD Cloud website up, right? So this is really great. And also we have this mass list, right? Right now, we don't have a structure. Yeah, our colleague in Bremen work on it, try to put a structure there. So this is just over the time, we saw those common inel compound. We said, okay, let's make a list. So that's how it's come from. And also you can do chem spider search. You select your data source, whichever you want, and then, give you the list. This is the website for this software, right? Simply called mycompounddiscover.com, right? From here, you can download the demo version, free demo version for 60 days. And you can watch the tutorial video, learn how to use it, and you can put your suggestion, complaint, or compliment, right? So our wonderful team will get back to you, very quick. I have to mention this quantification capability of a Q-Exactive, right? We know this. This is, one of the antioxidant, right? Simply, we just made the solution and run pulse switching, make sure, the mass is right, sure enough, right? We got a very good mass accuracy. And then this is the positive. We use the ammonium ion, right? We get excellent, linearity. This is the 3 triplet injection, and then the CV and diff are exceptionally good, right? And the same for the negative ion mode. So with that, I'm going to hand this back to Kai.
So hopefully we've been able to show you some of the capabilities for really identifying these extractables to leave you with no unknowns in your reports. We've presented GC and LC Orbitrap technology for this. We've also shown some empowering and new software workflows. As Kate mentioned, Compound Discover is available, Compound Discover 2, available and shipping in December. And Tracefinder is already out there and on sale. We've shown some online cloud-based libraries that HiChem are going to present in more detail today, but we've just touched upon those. And we also presented out the data from the ICP-MS. So before I close, and thank you for this, I just want to do a bit of promotion because, well, that's what we do. There was a great webinar last week from our chair here. If any of you want to catch it on demand, you can now at this URL, bit.ly /identify leachables. There's also another webinar that really deep dives into the GC Orbitrap and LC Orbitrap for leachables and extractables in December. You can register on the same website there. And if you need to find out any more information, as Kate said, it's on the mug, and we've actually over-ordered on the mug, so if anybody wants some outside, I don't want to take them home, I have a garage full, so please help yourself, and the URL on the mug, you can find a lot of great data. So with that, I'd just like to thank our collaborators who provided all the samples and did all of the work in the lab, and thank you all for your attention.